Sound check. So hello students, welcome to Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Now students, this is one short lecture on the chapter cell cycle and cell division. Now all of you might be aware of this chapter. This is much more a technical chapter than an easy chapter, right? Because you need to really understand and visualize this chapter rather than reading the lines of NCERT. Because let me tell you from my honest opinion, if you read the lines of NCERT line to line, there is a very high chance that you'll just mug it up and not understand the concept behind it. For example, what exactly happens in the case of mitosis, right? What exactly happens in the case of prophase, metaphase, anaphase, telophase? If you understand it visually, I am very sure you will never forget in your entire life. Let me give you a quick demo of what today's class is going to be, right? Let's see this chart, students. All of you, see this. This right here is a cell in the interface, right? This is a cell in the interface. Now, if we can see right now, a cell in the interface, all the chromosomes are in the form of a chromatin. Can you see, students? Entire thing is called as a chromatin. Can you see? Entire thing is in a chromatin stage. Now, this condensation of this chromatin happens in the next stage. So interface is a phase where the cell is in resting. Do not worry, if you don't know anything, I will teach you from the basics in today's class. This is just a visual representation of what we are going to achieve today. And let me tell you, students, all of you quickly smash the like button, because today's video is going to be the best lecture on cell cycle and cell division. That is my guarantee from my side, right? See, if you can see what exactly happens in the case of prophase, right? In the case of prophase, as you can see, all the chromosomes are condensed here, and then you can see the duplication of chromatid has happened. So let me tell you, students, this is going to be very interesting class, very interesting class. One by one, right? One by one, we'll be focusing on what exactly happens in the case of metaphase. If you see what exactly happens per chromosome. So all of this you will be learning in today's class. Complete one shot in 3D. So students, quickly let me know in the chat right now, are you ready for today's class? Quickly let me know in the chat, students. Quickly let me know. Are you ready for today's class? All of you, quickly. And all of you, quickly smash that like button. And all of you, right? Quickly smash that like button, all of you. Quickly. Done? Can we start, all of you? Can we start? Yes. Let me just check if my voice is proper. Right? Yes. So good evening, each and every one of you. Quickly smash that like button also. Quickly smash that like button, because it's very important for the YouTube algorithm. All of you know that. Now students, let me ask you a very simple thing. Right? Let me ask you a very simple thing, right? Has it ever happened to you that you get a cut? You get a cut on your hand and the entire cut gets healed on its own? Have you noticed that? The cut may be very fresh in the starting, but eventually the entire repair happens. Can anyone tell me how does it happen? What is the actual phenomena behind it? Now, for example, let me ask you one more thing. What will happen if I cut an entire arm? Tell me, students, what if will happen if we cut an entire arm? If you cut that entire arm, do you think it's going to grow back? All of you? See, a fingernail can heal up. A small cut can heal up. But do you think an entire arm can heal? Is it possible? Is it possible? Not possible. Especially in the case of humans, it is not possible. But have you noticed that? Have you noticed that? Is it possible in the case of a lizard? For example, if a tail of a lizard gets cut, the entire tail will grow back. The entire tail will grow back. It is possible there. It is possible there. Now, what if, students, if I ask you very simple question. The smallest cell or the first cell in the human body is a zygote. From a single zygote, we develop into an entire fetus. An entire fetus into an entire adult. How is it possible? For example, even in the case of a dog, right? This right here is a small Rottweiler puppy. Now, this puppy can grow into an entire adult. How is it possible? Anyone? How is it possible, students? How is it possible that one single cell or this small puppy can grow into a adult dog? What is the science behind it? What is the exactly the phenomena behind this mysterious process? The answer is very simple. The answer is name of the chapter, that is cell division. That is cell division. That is, from a single cell, a cell reproduces. That's right. A single cell reproduces and it divides and divides and divides, and eventually you get an entire dog. Entire dog eventually. Okay. Now students, what exactly happens in the case of cell division, students? In the case of cell division, a single zygote, students, listen to me very carefully, all of you. In the case of cell division, a single zygote, right, a single zygote can develop into around 3.6 trillion cells, or even more than that. It can divide and divide and divide. That is the magic behind it. That is the magic behind it. That is, a single zygote, students, undergoes a process. It undergoes a process called as cell division. Undergoes a process called as cell division. Now, if I ask you what exactly happens in the case of cell division, students, in the case of cell division, there are two main things that happen. In the case of cell division, there are two main things that are happening. That is, growth of a cell. Growth of a cell. Exactly, a cell is growing. At the same time, there is something called as reproduction. Reproduction. Now, what exactly is this reproduction? Reproduction here, meaning here is a single cell dividing into two. That is basic reproduction. So, a single zygote undergoes cell division. Now, students, I believe till here there is no doubt to any one of you, right? Basics is set, right? Now, if you want to do even more basics, let me teach you a few terminologies. Can you do that? Can we do, students? Can we learn a few basic terminologies right now? Can we do that? Right? Now, for example, what is the meaning of haploid? Like all of you might be knowing, right? All of you might know. What is haploid, students? Can anyone tell me? What is the meaning of haploid? Right? A cell or organism, cell or organism, right? Cell or organisms that has a single set of chromosome, a single set of chromosome. We call them haploid. Now, how do we represent haploid, students? Haploid is represented by N. Now, for example, in the case of humans, our diploid cells is 46 chromosomes. If a diploid is 46, what do you think is haploid content? If 2n is gonna be 46, what is n here, students? Can anyone tell me in the chat? The n is gonna be 23. The n is gonna be 23 in the case of haploid. Now, what is diploid? Diploid is again very simple, students. In the case of diploid, a cell or an organism has a pair of chromosome. Pair of chromosome. They have a pair of chromosome. Now, how do we represent the, students? We represent it by with the help of a symbol that is 2-1. Okay. Now, what is chromatin? You might have heard chromatin. Because in this chapter, I'll be using this word called as chromatin also very often. Now, what is chromatin? Chromatin is basically, students, a thread-like structure. It is a thread-like structure. It is a thread-like structure, students. What is it made up of? This thread-like structure is made up of DNA. It has our DNA plus it has something called as histone and non-histone proteins, as well as non-histone proteins. Now, this basic information, everyone should know what exactly is a chromatin. Now, what is a chromosome? Chromosome is a structure which has the DNA. That is, this chromatin, students, the thread-like structure. This thread-like structure will completely condense. For example, imagine there's a long thread. If you take this long thread and you make it into a ball and you give it a shape, A long thread, you make it into a ball and you give it a shape. That is a chromosome. That is a condensed chromatid. Now, for example, let me draw a simple chromosome right here. This is a simple chromosome, students. Can anyone tell me, students, in this chromosome, what is a chromatid? Can you see, students, this one? This right structures. These structures right here, 1, 2. These are called as sister chromatids. They are called as what, students? They are called as sister chromatid. Sister chromatid. Don't get confused. This is a chromatid. This is chromatin. Chromatin is a thread-like structure. This individual one is called as a chromatid. Now, what is this middle part? Middle part is called as centromere. It's called as centromere. Okay, students. Now, students, around the centromere, there is a cap-like structure. Can anyone tell me what is that? This cap-like structure right here is called as kinetochore. It is called as what, students? Kinetochore. Kinetochore. Okay. So this is a chromosome. A chromosome has how many chromatids? Can anyone tell me? A chromosome has 2 sister chromatids. It has a centromere. It has a kinetochore. Kinetochore. It also has a satellite. It also has telomeres, but that we have already discussed in the previous class. Clear? Now, I believe this basic information is clear to all of you. Well and good. Okay. Now, students, what exactly cell cycle now? Is cell cycle and cell division same to same? The answer is no. Because cell division is a part of cell cycle. Now, for example, a movie has action scenes, romance scenes. It also has some sad scenes. Movie is the entire thing, right? Cell cycle is the entire movie. A movie has multiple things in it, multiple different segments. For example, let's say the first segment of a cell cycle is called as cell growth. It is called as cell growth. In the case of cell growth, obviously, students, what is happening, students? Can anyone tell me in the chat? In the case of cell growth, there is increase in the cytoplasm. There is increase in the cytoplasm. Okay, done. Now, the second thing is going to be DNA replication. DNA replication. DNA replication. Now, what exactly happens in the case of DNA replication? In the case of DNA replication, for example, students, all of you, please focus here. If this is a chromosome, if this is the chromosome right here, and the DNA content here is 2C, let's say 2C, okay. Now, this individual single chromosome, can you see? It is going to become something like this. Now, this right here is called as 2C will become 4C. Listen to my words very carefully. When I say DNA replication, right, there is increase in DNA content, but there is no increase in chromosome number. Chromosome number will remain the same to same. For example, let's say here I have 46 chromosomes. Here also I will have 46 chromosomes. Chromosome number is going to remain the same. Chromosome number is going to remain the same. Then what is going to be the difference then? The difference is going to be the number of chromatids. For example, students, here every single chromosome has 1 chromatid. So total, tell me, students, totally how many chromatids do you notice here in the case of first diagram? Diagram number A, tell me the number of chromatids here. Number of chromatids. If 46 chromosomes are here, it will have 46 chromatids. 46 chromatids here. Okay. Now, what about in the second case? What about in the case of second case, students? Tell me in the chat quickly. In the case of second case, every single chromosome has 2 chromatids. So, what is the number of chromatids, students? Tell me the total number of chromatids in the case B. In the case of B, students, tell me the number of chromatids quickly in the chat, all of you. The number of chromatids which you would notice in the case of case B, 46 into 2. 46 into 2, students. Quickly tell me in the chat. And all of you, quickly smash that like button. I'm looking at the chat right now. Session PDF, I will upload it today only. Actually, I have given it from my end. It has to be uploaded, that's all. I have given it from my end. Don't worry. Tell me, students, the total number of chromatids would you see in the case B? See, here we have 46. Every single chromosome has a single chromatid. Okay. Now, what about here? What is 46 into 2, students? Tell me in the chat quickly. 46 into 2, quickly, all of you, quickly. Quickly, students. I can't even do multiplication now. Here we will notice 92 chromatids. 92 chromatids. Okay, 92 chromatids. Okay, now listen to me very carefully now. Third part is the most important part. Cell growth, DNA replication. Replication is happening. 2C to 4C. 2C to 4C is happening. And the third main part is the cell division. So cell division is technically part of cell cycle. It is technically part of cell cycle. Is that point clear to all of you, students? Is this point clear to all of you? If this is clear to all of you, quickly some fire emoji in the chat to let me know that yes sir, all is clear till now. All is clear. So cell cycle has three parts: cell growth, DNA replication, and cell division. Three main parts. Simple as it gets. Okay, simple as it gets. Now here is a small definition for all of you. Let me read for you. A sequence of events by which cell duplicates its genome, synthesis the other cell constituents, that is endoplasmic reticulum. It could be Golgi apparatus. It could be nucleus. It could be mitochondria. All of those. And eventually divides into two daughter cells is called a cell cycle. That is, from one cell we are getting two cells. Two cells. That is normal cell division. Normal cell division. Okay. Now students. Now let's understand something called as the phases of cell cycle. Now tell me students, how many of you love movies here? Raise your hands. Quickly raise your hands. How many of you love watching movies? And also tell me which is your favorite movie. Okay. Tell me. Now understand phase of cell cycle. Very, very easy to understand. Just follow me. Okay. Now the entire cell cycle is broadly classified into two categories. Okay. It is broadly classified into two categories, students. It is broadly classified into two separate categories. Okay. Into two categories. Okay. All of you listen to me very carefully now. The first category here is called as interphase. Interphase. Is that clear to all of you? Interphase. Okay. The second category is called as the M phase. It is called as the M phase. Okay. Now this interphase is eventually divided into three sub-stages. That is G1, S and G2. Don't worry if you don't know any one of them. I will be explaining to you in today's class, so do not worry at all. Okay. Now, what is M phase divided into? M phase is further divided into two separate categories. Okay. The first category is called as chirokinases. It's called as chirokinases. The second category is called as cytokinases. Cytokinases. Okay. Now, what exactly is chirokinases? The meaning of chirokinases. Chiro means nucleus. Chiro means nucleus. Kinases means division. Cyto means cytoplasm. Kinases means division. So, division of cytoplasm, division of nucleus. Division of nucleus and division of cytoplasm. Okay. Now, this chirokinases is further divided into something called as PMAT. PMAT. That is called as PMAT. It is called as PMAT. Now, what is PMAT, students? What does the PMAT stand for? The P stands for prophase. Prophase. What is M? M stands for metaphase. M stands for metaphase. A stands for anaphase. Anaphase. Now, what is T? T stands for telophase. Telophase. So, let me recap one more time to all of you what exactly is happening here. The entire cell cycle is divided into two separate categories. The first one is called as interphase, second is called as the M phase. Interphase is divided into G1, S, G2. M phase divided into chirokinesis, cytokinesis. Chirokinesis has PMAT, prophase, metaphase, anaphase, telophase. While cytokinesis basically has two parts again, students. Remember all of you, cytokinesis is different in the case of animals. It is different in the case of animal cells and different in the case of plant cell. Okay, animal cell and plant cell, it is going to be different. Why? Because plant cell is more strict, caduce it is. It has something called a cell wall, very difficult to do. Okay, so believe students, I believe till here all of you are very clear. It is very clear to all of you. Okay, now students, let me teach you a small concept called as time duration. Okay, understand. Now before we go any further, let me make the concept even more clear to you right now, more clear concept. Can all of you see this diagram? Is the diagram clear to all of you? It's clear to all of you. Okay. Now this entire thing is a cycle. This entire thing is the cell cycle. Entire thing is called as the cell cycle. Okay. Now students, can all of you notice this light blue region here? Can all of you notice this light blue region? This entire light blue region. This entire light blue region is called as what? It is called as what, students? This entire light blue region is called as interphase. It is called as interphase. Can you see? This entire light blue region is called as interphase. Now, do you notice here? A interphase, this entire thing has something called as G1, S, G2. So, G1, S, G2 is present right here. G1, S, G2. Entire thing is called as the interphase. This entire thing. Interphase. Now, students, do you see this entire black color part? Can anyone tell me what is the black color part here? All of you? That is nothing but the M phase. Can you see? Entire black color part is called as the M phase. Now listen to me very carefully. I told you the entire M phase divided into chirokinesis and cytokinesis. Yes or no? Yes. Now do you notice here? This entire light dark purple region is called as the chirokinesis because it has PMAT, prophase, metaphase, anaphase, telophase. And this light pink region, or what do you call it? Saffron color, right? Or it's a salmon color or something. This entire thing is the cytokinesis. Cytokinesis. Clear, students? This entire thing is called as a cell cycle. Now listen to me very carefully. How much time duration that each cell division takes place? For example, all of you, this entire cell cycle, this entire cell cycle can get over. It can get over in the case of bacterial cell. In the case of bacterial cell, the time duration required is just around 20 minutes. 20 minutes, the entire cell cycle is over. But what about in the case of yeast? It takes more time. Yeast is much more complex. Yeast takes around 90 minutes. 90 minutes. What about in the case of a human cell? A general human cell. A general human cell, not RBC, no, don't take RBC. Why? RBC doesn't have nucleus only. Okay, so remember that. You can't take RBC in all here. Okay, so general human cell takes around 24 hours. It takes 24 hours. 24 hours. Now let me tell you a small perspective here how this 24 hours is divided into. Now students, how many of you recently went for movies? How many of you recently went for any movie? Now generally what happens, I'm talking about movies in the theater, not in the house. Movies to a theater. When you go to a theatre, students, have you noticed that a movie will take place? Always there is a movie will go. Then we have a, let's say we have 1.5 hours movie. There will be a 10 minutes interval. There is a 10 minutes interval. 10 minutes interval. And again for 1.5 hours, let's say generally in 3 hours movie, 1.5 hours is the movie. Okay. Clear? This is a general movie. Okay. Now here, you need to understand here is that in the case of movies, you're watching for 1.5 hours, you're watching something, and 10 minutes is the interval where you're chilling little. Aram se chilling. Okay, in the interval. Okay. But in the case of cell division, it is the complete opposite. For example, in the case of cell division, this entire 1.5 hours is going to be something like a cell is going to be in interface. This small 10 minutes is equivalent to M phase, and again we have a long interface. Now tell me in the chat, students, quickly in the chat, in the cell cycle, in a general cell cycle, which stage is longer? In a general cell cycle, which stage is longer? Is it a M phase or is it interphase? Which stage is going to be longer here? Obviously, the longer phase is going to be the interphase. And in the case of human cell, that is these 24 hours is divided into 23 hours and 1 hour. This 23 hour is the interface. So, or this 24 hours in cell cycle, or this 24 hours in the entire cell cycle, 23 hours is in the interface, also called as resting phase. Okay, and this 1 hour is M phase. 1 hour is going to be the M phase. Let me tell you one more perspective. That is, this 23 hours, approximately 95% of the time, it is in interface, it is just resting. And this 1 hour, that is approximately 5% of the time, it is in the M phase. Is this easy to understand? Yes or no? Tell me, students. Yes or no? So, whole takeaway concept. The takeaway concept should be, in a cell cycle, interface is there, M phase is there. The longer time duration is for interface, less time duration is for the M phase. Is that takeaway good enough for all of you? Did all of you understand the basic concept of cell cycle? Which takes more time? Interface takes more time. That's all. Now, students, now we come to the different stages or different parts of your interface, also called as resting phase. Okay. Now, students, tell me, in movie interval, do you simply keep quiet or do you run to the washroom? Do you go buy popcorn? Do you buy Coca-Cola? Do you do something? No. Or you move around a little bit, throw some 3D goggles here and there. You throw around. You do something in the interval time. Here also, let me give you one more example. Okay, let me give you a simple example. All of you are preparing for NEET examination. Yes or no? Tell me in the chat quickly, all of you. All of you are preparing for NEET examination. Okay, all of you are preparing for NEET examination. Examination is coming up. Now, are you wasting your time or are you preparing for the examination? You're preparing for the examination. Imagine your entire preparation journey, including your exam, is like a cell cycle. It is like a cell cycle, right? You are right now preparing for the examination. Exam is in May, right? May 5th is the examination. That examination day is called as the M phase. That is called as the M phase. If that is called as the M phase, what do you call your phase right now? Can anyone tell me? For example, if you take your life as a cell cycle, exam day is the M phase where you go write the exam, where the division happens, very short duration, because exam is going to be there for only 1 day. Exam is going to be there for only 1 day, just like M phase, only 1 hour. Okay. The entire journey what you are going through right now, that is called as the interface. So all of you right now, where students, all of you right now in the all of you right now is in the interface where you're preparing for the examination. So just like how you are preparing for the examination, the cell also is preparing for the entire M phase. So let's check out the 3 different stages in the preparation. Shall we now? Shall we students? Shall we look at the different preparation stages? Right. Now all of you need to understand here is the first phase is called as the G1. The first phase is called as G1. Now this G1 phase is also called as first growth phase or gap 1 phase or first growth phase or post mitotic phase. Now you might ask me, why is it called as post mitotic? Ravindrani, Meghana, yes. Karthik, Amrita, yes. All of you agree? Why do I call it post mitotic? Why do I call it post mitotic? Listen to me. This entire thing is cell cycle. Now, so all of you notice here, G1, S, G2. After G2, what do we have? After G2, we have the M. After G2, I have the M here. Now, what do I have after M? Can anyone tell me? Can anyone tell me what do I have after M here? Anyone? After M, I have G1 again. Again, I have G1. So, can I call it post mitotic? Post M. After M, we have G1. After M, we have G1. All of you agree? So, it's called as post mitotic. After mitotic. Post means after. After mitotic is called as the M phase. Done. Okay. So, what exactly happens, students? What exactly happens here? Okay. What exactly happens with G1? Okay. First of all, understand, students, this is actually the longest phase. It is actually the longest phase. It is actually the longest phase in the entire cell cycle. Longest phase in the entire cell cycle. Okay. Now, this phase occurs between. It occurs between. Can anyone tell me? It occurs between which 2 stages? Can anyone tell me in between which 2 stages do we find G1? We find it between, students. We find it between M and S. Can all of you agree? It is occurring between M phase and S phase. Here, S is here. Done. Now, what exactly happens here, students? Understand. Okay. The cell here is technically metabolically active. It is going to be metabolically active. It is metabolically active, and it is going to continuously. It is going to continuously grow, but is very important. The cell is going to continuously grow, but does not replicate DNA. It does not replicate DNA. That is, the cell is metabolically active. It is continuously growing. The cell is growing here, but does not replicate DNA. So if someone asks you, does the DNA replication occur in the case of G1? What is your answer? Tell me in the chat right now. All of you. Does the DNA replication happens here? Does not happen. So where does it happen? Where does the replication of DNA happens? The entire DNA replication, students, the entire DNA replication happens in the S phase. Happens in the S phase. That is, first point here is DNA replication. DNA replication occurs here. That is, if a cell is 2C, it is going to become 4C. 2C to 4C conversion happens. That is, if this is the chromosome like this, this becomes something like this. Clear? A single chromatid to double chromatid. That is the first main point here. First main point. The second main point is about the centrioles. Is the centrioles. The centrioles also duplicate. They also duplicate. Now students, understand here, DNA replication, where do you think happens? Does it happen in the cytoplasm or does it happen in the case of nucleus? It happens in the case of nucleus, obviously. It occurs in the nucleus. While central duplication occurs in the cytoplasm. It occurs in the cytoplasm. Now this much is given NCERT. Let me write one more point for all of you. That is, in the case of S phase, right? In the case of S phase, the most important point, okay, let me use a different color. No change in chromosome number. This right here is the basics of many numericals. Is everyone following here? There is increase in DNA content. There is increase in DNA content, but the DNA chromosome number, it is gonna remain as such. That is, if there were 46 chromosomes, it is gonna remain 46 itself. The number of chromosome is not gonna change at all. The number of chromosomes is gonna remain the same, but the number of chromatid might vary. It will vary. Is that point clear to all of you? Very basic information I'm giving you today. Very basic information. Clear, students? Clear? Now, students, download these notes. I will make sure it's going to be available to you, right? So download them. Okay? Now, next point is going to be very important. Again, G2 students. G2 is also very important. Now, students, what if I call G2 as, it can be called as second growth phase. All of you agree with me? Second growth phase. All of you agree with me. Can I also call it pre-mitotic phase? Pre-mitotic phase. That is, this is a phase. G2 is a phase which is occurring where? Before M phase. So can I call G2? Can I call G2 or the gap 2 phase as pre-mitotic phase? Pre-mitotic phase. Done. Pre-mitotic phase. Beautiful. Pre-mitotic phase. Now what exactly happens, students? What happens? Okay. Here in this case there is duplication of mitochondria. Duplication of mitochondria and chloroplast occurs here. Okay. Apart from that, with the question which will be asked in numericals, I'm telling you, question will be asked mainly based on this last segment, that is, most of the RNA synthesis and protein synthesis happens here. Protein synthesis occurs here. Remember, students, even in G1 also it happens. RNA protein synthesis even in G1 it is happening, but majority it is happening in the G2 phase. Is that clear to all of you? Gap 2 phase. Gap 2. Is that point clear to all of you? With that, the entire cell cycle is over actually. With that, the entire cell cycle is over. This entire basics is over. Okay. Why is this here? This is not supposed to be here. This is supposed to be later on. Okay. Now, students, do you ever get fed up? Do you ever get fed up that you don't want to do anything anymore and you're so fed up that you just want to go sleep and not do anything? Does it ever happen to you? In your preparation, does it ever happen that at one point you're so done, you're so burnt out, you just want to go temporarily sleep? Does it ever happen? It does happen. It happens to everyone. If you're a human, it does happen to you, right? Same thing happens in the case of cell also. Some cells, right? Some cells are so fed up, right? They're not technically fed up because they do not have lack of nutrients or they do not have a specific role that time, right? Some cells, they leave the cell cycle. That is, after G1, they exit the cell cycle and they go into a temporary arrest. They're like, oh, namaskar. They'll go temporarily, they're going to arrest. They'll become temporary, or in some cases, permanently, they'll become arrested. And they will never finish cell cycle. They don't want to divide. They don't want to divide at all. They're so stubborn. Now that stage, right, a stubborn stage, not the teenage, okay, their teenage is called as the G0 phase. It is called as what, students? G0 phase, also called as quiescent stage. Okay, quiescent stage. Very fancy, no? Yeah. Now, what is quiescent stage? Let me read the lines of NCERT. Very easily mentioned. Some cells in the adult animals do not appear to exhibit division. They're fed up. Okay, appear to division. Example is very important. Heart cell is the example. Heart cell and nerve cell is a simple example. And many other cells divide only occasionally as needed, replace other cells that have lost because of injury or death. Now these cells that do not divide further, exit G1. So when do they exit? They exit after G1 and enter the inactive stage called as quiescent stage or G0 stage of the cell cycle. Now students, the PYQ alert right here, PYQ alert, see here. Very important line. All of you highlight this, highlight it. Okay, I don't have a highlighter. What is this? What is this? Red flag, huh? Okay, red flag. We'll put red flag here. Okay. Red flag. Okay. Listen to me. Cells in this stage remain metabolically active. They are going to be metabolically active but no longer proliferate. Very important line. That is, the G0 cells, they are metabolically active. They're ready to go. They're like, I am waiting for my opportunity. Okay. But they don't divide. But are they active? Yes, they are active. Okay. Yes, they are active. Nice, no? Unless called upon, do so depending on the requirement of the cell. Now there is G0 temporarily re-enter G1 phase and undergo division. G0 permanent, never end divide. They never divide. Okay. Some cells temporarily arrested. Some cells are permanently arrested. Temporary, permanent. Okay. All right. Now students, here is a line of NCERT which is confusing to many students because this right here is called as foreshadowing. Okay. Now this lines here, right? These lines here are actually talking about other chapters information. For example, they're talking about the plant kingdom here. All of you focus. Now I learned something. Now I'm going to use it. Where's that? Where did I get that, students? Where is the flag coming from? Here, no? Okay, let's put some sun here. Very important, I guess. Sun one here and here. Okay, very important. Listen to me, all of you. Today, figure out something new. Okay. In animal cells, mitotic cell division is only seen in diploid cell. In the case of animal cell, students, mitosis is seen only in the case of diploid cell. That is, a diploid cell will undergo mitosis or okay, somatic cells, mitosis, somatic cells. However, there are few exceptions to this where a diploid cell, students, whereas a haploid cell, okay, haploid cell divides by mitosis. So generally what happens is in the case of our body, generally a diploid cell will undergo mitosis. But in the case of a plant cell, spores to form the sporophyte in the plant kingdom. Okay. Now this haploid cell can also undergo mitosis. Don't worry, even a haploid cell undergo mitosis. This you will learn in the plant kingdom. For your recollection of the examples of alternation generation in plant kingdom, identify the plant species that is mostly algae. Mostly algae undergoes, haploid cell undergoes mitosis. Remember that. Okay. Now students, a question to all of you. A question to all of you students. A question to all of you. Let's see who can tell this question. Let's see who can tell this answer. Students, smash the like button. What is this? Target is 200 today also. Target is 200. Target is 200 quickly. Yes, even B's also. Can you think of a cell that never enters G0 phase? These cells are, what do I say, special. They don't undergo G0. They keep on dividing all their life. Let me give a small clue. These cells are responsible for plant's height. These cells are responsible for plant's height. All of you quickly. These cells are responsible for plant height, like tall plant, no? Tall plant. Okay, 6 feet, 10 feet, 20 feet, 30 feet. How do they grow so tall? How do they grow so tall? Because of a cell called as meristematic cell. Because of a cell called as meristematic cells. Meristematic cells are the cells which do not undergo G0. They will keep on dividing and dividing and dividing. Okay, G0. Now students, one small concept if we go before we go further, right? This is called as a checkpoint, right? See, for example, if you're going in aeroplane, they'll check you nicely. If you're going to the metro, if you're going to the anywhere, they check nicely, you know, if you're not carrying some gun and all. In the case of cell cycles, there are checkpoints. Okay. First checkpoint is after G1. Can you see? First checkpoint is called as G1 checkpoint or a G1S checkpoint. G1S checkpoint. Okay. Now here, in G1S checkpoint, the cell will always make sure that enough of energy is there for replication. Will make sure the DNA is not damaged. If DNA is damaged, imagine if DNA is damaged, the damaged DNA will replicate. Problem then? Problem. Yes. So check for nutrients, growth factors and DNA damage. Nothing should be there. That is before S. Before S. Now one checkpoint is after G2. It is called as G2M. G2M checkpoint. Here they'll check for cell size. Is the cell size enough for cytokinesis later on? And to check the DNA replication has occurred properly or not. These two are the major checkpoints. After G1, after G2. Okay. Now students, there is one more checkpoint that happens after the metaphase. Okay. That is called as metaphase checkpoint. Here, chromosome spindle fiber attachment. In metaphase checkpoint, they will check if there is a proper attachment of chromosomes on the equatorial plate. That is metaphase checkpoint. Okay. Now, I believe till now there is no doubt to any one of you. Quickly, yes or no? No doubt till now. All of you, no doubt till now. We have learnt about the entire interface now. Interface is over. Entire interface is over right now. Because now we'll be starting something called as the entire M phase. All of you ready? All of you ready, students? Ready? Because the entire M phase has something called as chirokinesis, cytokinesis, which I already told you. And a chirokinesis mainly has prophase, metaphase, anaphase, telophase. Now, one by one, we'll be dealing each one of them. Are you ready, students? All of you drink some water. All of you drink some water, students. All of you drank water? Done? Water break over? Water break over, students? Now, students, tell me, is there any other chapter backlog in your class 11th? Do any one of you have any backlog in class 11th? Let me know in the chat box right now. I'll look at it. Okay. Any one of you? Any backlog in class 11, students? Any backlog in class 11th? All of you? 200 ml of water. Nice. Drink. Before I start, prophase. Now, all of you students, quickly smash the like button. And I was reading the comments, right? What is IPE session? I don't know what is IPE. Okay. Structural organization. That is not my forte. If to ask Gopika ma'am to take it one more time. Don't worry, we'll ask Gopika ma'am. Okay. If not, I will also I'll try taking it. Anatomy, anatomy is so easy. Anatomy is so easy. Anatomy. Don't worry. Next week, this week itself actually. This week we'll be doing. Do you want to do anatomy or plant growth regulators? Quickly tell me in the chat. Okay. Anatomy or plant growth regulators? Let me know in the chat. Let me know in the chat. Okay. I was thinking plant growth. Let's see. Now, students, let's start with something called as prophase now. Okay. Can we start? See here. Chirokinesis, PMAT, prophase, metaphase, anaphase, telophase. Okay. Prophase, metaphase, telophase, anaphase. Inter-public exam, IPE. Oh, IPE. Now it makes sense, but no. Okay. Now, students, understand what is prophase. Now, I'll make you write the notes. So, I want everyone to write the notes with me. Otherwise, I could just add some image. So, I'm going to write the notes. I'm going to draw for you. All of you follow through. Okay. Now, what exactly happens in the case of prophase? Understand. First of all, it is the longest phase. It is the prophase is the longest phase. It is the longest phase. Done. Longest phase of M. M phase. Longest phase of M phase. Okay. Done. Now what happens here? In the case of M phase, there is initiation. There is initiation of condensation of chromosome. Condensation of chromosomal material. Initiation of chromosomal material. Basically, the entire chromatin undergoes condensation. Entire chromatin undergoes condensation. Okay. Entire chromatin undergoes condensation. Okay. Now, entire prophase can be broadly classified into two separate categories. Okay. Entire prophase can be classified into two separate categories. The first one is called as early prophase. Early prophase. Obviously, what is the opposite of early? Early and late prophase. Late prophase. Early and late prophase. Okay, early and late prophase. Now, what exactly happens in each one of them? In the case of early prophase, okay, there is condensation of chromosome. There is condensation of chromosome. Apart from condensation of chromosome, there is movement of centrosome. There is movement of centrosome. I believe all of you remember centrosomes. For example, let me draw and show you. Let's just say this is our cell right here. This right here is the cell. Okay, this right here is the cell here. Okay, small cell here. Now what happens here? Understand all of you. In the middle we have the nucleus. In the nucleus, all the chromatins are started to condense here. They have started to condense here. Now apart from this, towards the corners, we have the centrosome. Can you see all of you centrosome? And every centrosome has something called, students, what does it have? It has centrioles. How many centrioles? 2 centrioles. So one centrosome has 2 centrioles. All of you should remember. That 2 centrioles. Now, students, around the centrosome, there is pericentriolar space here. Okay. Now, from this pericentriolar space, there is formation of spindle fiber. Now, spindle fiber formation is here. On the back side also there is spindle fiber formation. On the back side also. Front side and back side. Okay. Now understand, these centrioles are producing two types of microtubules. Two types of microtubules. Two types of microtubules. Okay. Two types of microtubules here. The first type is called as this one right here, which is on the front. That is called as spindle fiber. That is called as spindle fiber. The first one. Now, what is in the back side, right? Back side. That is called as aster plus aster. Aster. Now, both of them are what? Both of them are a type of microtubules. Both of them are a type of microtubules. Each one of them has a different function. For example, spindle fiber will attach to the chromosomes, while asters will help in anchorage. They'll help in anchorage. So, two different types of microtubules arising from the centrioles or centrosome here. Now, understand very carefully. This aster plus spindle fiber, together they are called as mitotic apparatus. They are called as what, students? Mitotic apparatus. Together they are called as mitotic apparatus. Mitotic apparatus. They are called as mitotic apparatus. Clear, all of you? Is this clear? Yes, it is clear. Okay. Now, students, remember, extra point I'll tell you. The spindle fiber right here is found in the case of animal cell as well as plant cell, while asters are found only in the case of animal cell. They are found only in animals. Asters are found only in animal. Only in animal cell. Only in animal cell. Asters. Okay. Now, this entire thing is happening in the early prophase. Early prophase. Then what exactly happening in the case of late prophase? In the case of late prophase, there is a disappearing. People are going out. Okay. There is a disappearing. Who is disappearing, students? Other different organelles are disappearing. For example, Golgi complex, endoplasmic reticulum, nucleus is entire disappearing. Nuclear envelope mostly, right? Nuclear envelope is disappearing. Nucleolus is disappearing. Nucleolus, everything is disappearing. Everything is disappearing in the case of prophase. Now, all of you, I'll give you a small recap. Listen to me. Small recap. Okay. Prophase, longest phase of the M phase. Initiation of condensation of chromosome, that is chromatin material is getting condensed here. You see, chromatin material is getting condensed here. Compaction of chromatin material is happening. It is happening. Okay. Early prophase, late prophase. Early prophase, what is happening? Condensation of chromosome. Apart from condensation of chromosome, there is movement of centrosome. Can you see? These centrosomes are moved up here. Centrosomes are moved up here. Okay. Now, the centrosomes has centrioles. Centrioles has two types of microtubules. The first type here, can you see, all of you? This is called as spindle fiber. Is called as spindle fiber. And can you, this one is called as aster. Together they are called as mitotic apparatus. Together they are called as mitotic apparatus. Together. This is spindle fiber here. This is asters on the back. Together they are called as mitotic apparatus. Clear, all of you? Clear? Is that point clear to all of you? Is that point clear to all of you, students? Quickly tell me in the chat. Are those points clear to all of you? No, see here. Can all of you see this? This is the entire asters. They're moving to the opposite poles, movement of. Then from this particular centrioles, there is spindle fibers that are arising and asters are rising on the outside. Asters. This is called as the movement of chromosome. Movement of centrosome. Not chromosome, movement of centrosome. Okay, and there is compaction here. This compaction is happening here. It is happening. Okay, that is the entire prophase. That is the entire prophase. Students, is that clear to all of you? Is that clear to all of you, students? Clear? Can you see here? Early prophase, chromatin, late prophase, chromosomes. Condensation has occurred here. Condensation has occurred. Okay. Now, what exactly happens in the case of metaphase now? In the case of metaphase, very simple, very easy. Okay, metaphase, what happens? Let me draw the diagram and show you. Okay, metaphase, what happens? Okay, this is the cell right here. This is cell. Now, what is happening here is in the cell right here. Should I use orange, right? Okay. Now here, what do we have? We have the centrioles here. We have the centrioles. From the centriole, we had pericentriolar space, pericentriolar kinoplasm here. Spindle fibers are arising. Spindle fibers are rising. Now, what is happening in the case of metaphase is that all the chromosomes are aligning at the equatorial plate. There is something called as kinetochore here. Kinetochore. Can you see, all of you see that? Yes, you can. So all the, let's say butterfly, no? All the chromosomes are arranged at the equatorial plate. What is happening here? First of all, there is something called as movement of chromosome. The entire, there is movement of chromosome. Movement of chromosome, that is called as congregation. Okay. Now, this right here is called as metaphasic plate. Right here is called as metaphasic plate. Metaphasic plate, also called as equatorial plate. Equatorial plate. Okay. Now here we have the spindle fibers. The spindle fiber. Okay. Now students, understand here. Let me draw and show you one more time. For example, this is the chromosome. This is the chromosome right here. On this chromosome, there is kinetochore here. Kinetochore is here. Okay. Right. Kinetochore. Okay. Kinetochore. Now here, all the spindle fibers are attaching to the kinetochore. So please remember this. This is what is happening here. This is what is happening here. Your entire chromosomes, entire spindle fiber will never attach to centromere. Never to the centromere. It will always attach to the kinetochore. Kinetochore. Okay. Let me write for you, otherwise you'll forget off. Spindle fiber attached to the kinetochore. Spindle fibers attach to kinetochore. Please remember this. This right here is a NEET PYQ. I cannot stress enough. Here. To kinetochore. Done. Okay. Now students, here, how many in total of, how many chromosomes are there, students, tell me? If this is a cell right here, how many chromosomes are there? There are totally 46 chromosomes. Which has 92 chromatids. Please remember this also. 92 chromatids right here. 92 chromatids right here. Okay. Now students, understand here is every chromosome has spindle fiber attaching to how many sides? Is it both the sides or a single side? to both the sides. Now, why is it important? Because in the meiosis, it is going to be only one side. So remember, spindle fibers are attaching from the both the sides to one chromosome, okay, both the sides. And this is the best stage. It is the best stage to study chromosomal morphology. Best stage study chromosomal morphology, because condensation is complete here, because the entire chromosomes are condensed here. Why? Because condensation is complete. Here. Condensation is complete. Students, are these points all of you writing down? Yes or no? Tell me, yes or no? Right? Movement of chromosomes, metaphasic plate or equatorial plate, spindle fiber attached to kinetochore. Best stage to study the chromosome morphology, that is condensation is complete here. Condensation is complete. Now students, if you see here, right, all of you see, all the chromosomes are aligning to the center right here. All the chromosomes are center. That center part is called as metaphasic plate. Center is called as metaphasic plate or equatorial plate. Metaphase plate or equatorial plate, what do I call it? Okay. Now, next. Can you see all of you? This is actually the under the microscope, metaphasic plate. Okay. Transition to metaphase. Okay. Now this is called as chirogram, and this is basically done at the metaphase stage, because base stage study morphology. So study of morphology of the chromosomes is called as chirogram. This right here is a chirogram, and all of these chromosomes are at metaphase right now. Okay. Now anaphase. Ready? Anaphase is very simple, very easy. I'll tell you what you need to remember here and nothing more than that. Okay. Anaphase. Understand what exactly happens in the case of anaphase. Let me teach you again with the help of a diagram. Okay, with the help of a diagram only. Okay. Now this is your cell. Here we have the centrosomes, two centrioles on that. Okay. Now all of you need to follow very carefully. Now here this diagram is very important, students. If you understand the diagram, everything is going to be very easy to you. Some hard sum drawing, no? What exactly happened here? This was the chromosome. Right. This was the chromosome with the metaphase. For example, see this. I'll erase this later on, but see this. This was the metaphase, right? This was the metaphase chromosomes. This was the entire metaphase chromosome. Now what happens here is the entire centromere splits. This centromere is splitting. This centromere is splitting. This centromere is splitting. When that split is happening, the centromere is splitting apart, the chromatids are moving apart. Okay. So what happened here is the entire centromere split open. This was the, I'll draw one more diagram small here. If this was the entire chromosome, it split in half. It completely split in half, and both the sister chromatids became separate. Can you see? They became separate. This entire thing was at the plate. Now they are split open, and half of the chromatids are going this side, half of the chromatids are going upside. So half chromatids this side, half chromatids this side, on top to top. Okay, top and top. Now students, can anyone remind me again, how many chromosomes do we have here? All of you tell me. Write down the points before that. This is the shortest phase. Shortest phase. Okay. Sister chromatids, okay, of the chromosome split. They completely split here. Split. Okay. Now students, tell me, how many chromosomes do we have here? 46 chromosomes we had. 46 chromosomes. How many chromatids, can anyone remind me? How many chromatids? 92 chromatids. 92 chromatids in this stage. Now listen to me very carefully. The most important line of this particular chapter, I would say, right? If you want to understand the entire cell cycle, very important. Okay. Now here, all of you listen to me very carefully. Eyes open. Eyes, ears, everything open now. Very focus. Now this particular chromatid eventually be part of a new cell. It will be part of a new cell eventually. Yes or no? Yes. Now listen to me very carefully, all of you. Can I call this as daughter chromatid? This daughter chromatid. This daughter chromatid will act like daughter chromosome. Right? Daughter chromatid will act like a daughter chromosome. So what happened here is because it will be a daughter chromosome for future daughter nuclei. Okay. Now this entire chromatids, all the entire chromatids here will act like a chromosome. So all of these 92 chromatids are technically gonna act like chromosome. Temporarily. Now students, tell me, what is usually N represented as? N is 23. 2N is what, students? 2N is 46. All of you. 3N is going to be 69. 4N is going to be 92. So this cell right here, students, this cell right here is temporarily at a 4N condition. It is at a 4N condition temporarily. Why? Because every single daughter chromatid is acting like a daughter chromosomes temporarily. So this cell right here is at 4N condition temporarily. Is that point clear to all of you? Because I'll come back to this point here when I teach you DNA content. So this particular cell is in 4N condition right now. Temporarily 4N condition. Temporarily. Okay. Now 4N condition. Now can you see here? This direction also matters. This is the centromere. Centromere position is always towards the poles. Centromere position is towards the pole. Centromere position is towards the poles. Can you see here also? This is the centromere. It is always towards the pole. That is very important. It is also given your NCERT also. Okay. Now students, so what we learn here? We learn that in the case of anaphase, entire cells will be pulled apart. Can you see? It will be pulled apart here. They'll be pulled apart. And always remember, the centromere position is towards the poles. Always remember, centromere position is always towards the pole. That is very important. Okay. And moreover, it's not centromere, it's kinetochore. Actually, centromere and kinetochore also there. It is always towards the pole, and there is splitting of sister chromatids. All the sister chromatids are split open. They are splitting open. Okay, done. Now, students, telophase is very simple again. Telophase, nothing. Oh, one second, students. Last point here. I forgot to mention. This is the best stage to study shape of chromosome. Best stage. That is V shape, metacentric, submetacentric, telocentric, right? Telocentric, acrocentric. It is best stage study. Best stage to study the shape of the chromosome, anaphase. Okay. Now, what about telophase? Telophase is very simple. It is the complete opposite of anaphase. Because complete opposite of prophase. Complete opposite of prophase. Everything is reappearing here. Okay. I'll draw my diagram only, it's better. Can you see, students? All the chromatids are here. And remember, always single chromatid now. They're always single chromatid here. Please remember that, because splitting has happened now. Okay. Splitting has occurred now. So this is called as telophase. Okay. That is, chromosomes cluster at the opposite poles. Chromosomes will cluster at the opposite poles. Now, when this is happening, okay, all the chromosomes will again turn back to chromatid. The shape of the chromosome is lost. That is called as loss of identity. Okay. Loss of identity is happening. Every other thing is developing back. Nuclear envelope is developing back. Will develop. There will be Golgi complex, endoplasmic reticulum. Everything will reform. Nucleus, everything will reform at the telophase. Everything will be reforming. Okay. That is the entire telophase. Can you see here? This is the telophase. This is it's called as contractile ring, this entire thing. Will be pulled apart. Chromosomes are pulled to the cluster towards the opposite poles. And this is the end of mitosis. Telophase. Okay. So anaphase being pulled apart, telophase is the end. Okay. Now students, we learned right now chirokinesis, prophase, metaphase, anaphase, telophase, all done. Okay. Now we come to something called as cytokinesis. Before I go to cytokinesis, let me give you an entire recap of mitosis till now what we learned. Okay, till now what did we learn? All of you, focus. We learned about prophase, longest phase, initiation contents of chromosomes, early, late. Early prophase, what is happening? Late prophase, what is happening? Spindle fibers as together, they're called as mitotic apparatus. Okay. Then we learned about metaphase, metaphasic plate, movement of chromosomes, spindle fiber attached to the kinetochore. Best stage to study the chromosome morphology. Then we learned about anaphase, shortest phase, sister chromatids of the split. Sister chromatids split. Okay, they will completely split open. Split. Daughter chromosome will act like chromosome. So in total we have 92 chromosomes here. 92 chromosomes in this here. 92 chromosomes. Best is study the chromosome shape. Telophase, all the chromosomes align, cluster at the opposite poles. Everything, they lose their identity. Everything is reforming. Everything is reforming here. Everything is reforming. And that is the end of mitosis. That is the end of mitosis. Now, what is cytokinesis? Cytokinesis is basically division of cytoplasm. That's all. Division of cytoplasm. Okay. Now, cytokinesis is different in the case of plant cell and animal cell. It is different. Very different from each other. How is it different? Understand. Okay. Now, in the case of plant cell and animal cell, it is different. Let me give you a small example for animal cell right now. Now, imagine you have a clay. All of you know what is clay or balloon. Let's take a balloon itself. All of you know balloon. Imagine this is a balloon right here. Now, imagine what if you start pinching from both the sides. If you're pinching from the sides, from outside, if you keep pinching, eventually the balloon will meet in the end. In the middle, it will meet. Or if you have a clay, if you keep pinching the clay from both the sides, eventually you will meet. That is called as furrow formation. So if you see in the case of animal cell, if this is the cell right here, this cell will start to form something called as furrows. Can all of you see this? Can all of you see here? This is called as furrow formation. Here, furrow formation is happening. Okay. Here, furrow formation is happening. Furrow formation. So if you keep pinching it, eventually it will become two cells. Eventually become two cells. And this is basically centripetal in manner. That is furrow formation. Now, in the case of plant cell, that you cannot pinch from inside. Not possible. In the case of plant cell, it is not possible to just squeeze from outside. Why? Because in the case of plant cell, there is a hard layer on the outside called as cell wall. So if the cell wall is present, you cannot pinch at all. Not possible to, it will not bulge up. Like, I will not care. Okay. So in that condition, I have always told you, if your mother cannot make you listen from the outside, how much ever she will hit you, nice, pat. Eventually you will become immune. How much will hit me more? You will tell, I don't care. So what does your mother do that time? Your mother will emotional blackmail you. True or false? I hope my mother doesn't listen to it. So if your mother is not able to bulge you from outside, there will be emotional blackmail from the inside now. Emotional blackmail. That is what we are doing here. Since we cannot go from outside, what do we do? Since we are not going from outside, emotionally on the inside will split open. Emotional blackmail will do. Okay. So what is happening here is, can you see here? In the middle, on the inside, there is something called as cell plate formation. The cell plate formation is basically because of phragmoplast. That is, Golgi complex is involved here. Golgi complex, extra information, no need. Okay. Remember, there's something called as Golgi complex involvement is here. Okay. And from inside, now this will start to move outwards. That is called as cell plate formation. From the inside it will form, and this will move this side, outwards. That is called a cell plate. And this is centripetal, this is centrifugal. In manner. Centrifugal. Okay. Centrifugal in nature. Okay. Not furrow. Okay. I think this is ulta, it's written here. Okay. Let's erase this. Okay. So cell plate formation, furrow formation. Done, students? Done, all of you? Done? Done, students? All of you understood? Yes or no? If you understood, quickly smash it like button. 100 likes only is not done still. What is this? Centrifugal. Now, what is the significance? Growth and development, yes. Repair and maintenance, yes. Growth in meristem, restoring nucleocytoplasmic ratio. That is, imagine if the cytoplasm keeps on increasing, but the nucleus is not dividing. Imbalance, to correct that imbalance, mitosis is there. And that is in unicellular organisms. In unicellular organisms. Okay. Now, students, one question to all of you. Quickly go tell me in the chat. How many of you can tell me the answer to this question? Extra information for all of you today. Extra information because I'm happy. Okay. Where mitosis happened? What do you mean by that? Inside the cell only, right? Tell me. Did you find it? The entire cell only divides. Anyone found? Did anyone find centriole? Did anyone find centriole here? Tell me, where do you find? Do you see anywhere? Do you see centriole anywhere? No. Centriole is absent. Centriole is absent. If centriole is absent here, how do you think the entire process takes place? For example, if there is no centriole, from where does this assemblage of spindle fibers arise? Anyone? Anyone? If there is no centriole, how does spindle fibers even come into existence here? The answer is very simple. Very simple. It's not given in NCERT, but it should have been given. Okay. There is something called as microtubule organizing center. MTOC. Microtubule organizing center. Now this microtubule organizing center will make sure all the spindle fibers are attached and everything. Okay. An area present just near the nuclear membrane. Just near the nuclear membrane called as MTOC can serve as organize and assemble the microtubules required for spindle fiber formation. So spindle fiber formation in the case of plant cell is done by MTOC, microtubule organization center. Clear? Organizing center. All of you learned something new today? Yes, all of you learned something new today. Okay. Now, if you learned something new today, let me tell you one more extra information. Okay. This entire cell cycle, students, entire cell cycle is mainly dependent on two main factors. Something called as cyclins and CDK, cyclin dependent kinase. Cyclin dependent kinase. Now these two factors, right? These two factors completely control the cell cycle. For example, there are multiple types of your cyclins. There is S phase cyclin, there is G1 phase cyclin, G2 phase cyclin. Okay, so cyclins and CDKs completely control the entire cell cycle. Again, extra information for you today. Okay. Okay. This I think I already mentioned to you, right? If spindle fibers are not attached properly, the cycle is paused until the chromatids are separated attached to the spindle fiber. That is, just after metaphase. Just after metaphase, if all the spindle fibers are not attached properly, then what happens? Okay. A protein called as anaphase promoting complex APC. Anaphase protein promoting complex helps the cell to proceed to anaphase. So what happens is in some cells the metaphase does not go very smoothly, right? After metaphase the anaphase doesn't come itself. So in certain cases APC is released. APC is released and this protein helps into transition to the anaphase. Remember that. Okay, this is before checkpoint. Okay. Now students, tell me what will happen now. Tell me what will happen now. What if there is no cytokinesis and only cheirokinesis? Do you think something's going to happen that time? Do you think something will happen? Students, what is if there is no cytokinesis, only cheirokinesis? Only cheirokinesis always. Do you think anything will happen that time? Anyone? Anyone? No cytokinesis. The cell is not dividing only. The cell is not dividing. The nucleus keep on dividing. In such cases, there is a multinuclear condition. Yes, multinuclear condition will arise. Such multinuclear condition is called as syncytium. Okay, or coconut water. Okay, there is formation of syncytium. Cheirokinesis not followed by cytokinesis. That leads to multinuclear condition. That is formation of syncytium. Liquid endosperm of coconut is an example. Is an example. Clear, all of you? Clear? Yes or no? No. Students, now you tell me, are you ready to revise this entire thing in one more time in a DNA content wise? I always teach this to my students to get a better clarity. Now, if you are my student and if you have learned this chapter from me before, this should be an easy thing for you. Okay. Now, for example, in G1 phase, what did we have? We had a single chromomer like this. Okay. Let's just say this is 2 and 2C. 2 and 2C. G1. Now, what happened in the case of S? In the case of S, students, what happened? Was there a change in DNA content? Yes, sir. The DNA content doubled in the case of S. So this became, number of chromosomes is going to remain the same. DNA content became 4C. So in the case of S phase, it was 2 and 4C. 2 and 4C. Okay, 2 and 4C. Okay. Now what about in the case of G2? Will there be any change? Absolutely not. There is no change in G2. Okay, no change in D2. It's going to remain as 2 and 4C. Okay. Now, what about in the case of prophase? Is there any change in the case of prophase, students? Absolutely not. In the case of prophase, it's going to again remain 2 and 4C. Same goes for meta. Same goes for metaphase. There's no change in metaphase also. Metaphase also. Now, what happens in the case of anaphase? In the case of anaphase, there is split of centromere. The centromere is splitting. Centromere is splitting. Centromere is splitting here. Obviously, centromere is splitting. Now, I told you, in the case of anaphase, do all of you remember? In the case of anaphase, the cell was temporarily, what stage, students? Foreign. Temporarily foreign. Yes, all of you remember. Temporarily, in the case of anaphase, the cell was in foreign condition. The cell was in foreign condition. So this right here is 4N 4C condition. 4N 4C condition. Done. Now what about in the case of telophase? In the case of telophase, there are two things which can happen. Per cell, per nuclei. Okay. Per nuclei and per cell. The entire cell is going to be 2N. Entire cell is going to be 4N 4C. Okay. Per nuclei, it is going to be 2N 2C. Why? Understand with me. Understand why 2N 2C. Let me just go back and show you. Here. Now, this is your entire telophase. The entire cell is 4N 4C fresh out of your anaphase. Now, out of this 4N, can I tell 2N is going this side and 2N is going this side? Half of it is dividing. Half the number of chromosomes are coming this side. Half the number of chromosomes are going up. Half of division number of chromosomes? Yes. Half of the DNA content is this side. Half of the DNA content is this side. So what do we have? Entire cell right now is 4 and 4C. But this per nuclei, this side nuclei is 2 and 2C. This side nuclei is 2 and 2C. 2 and 2C. Okay. Now students, all of you focus. With what did we start, students? 2 and 2C we started. What are we finishing with? 2 and 2C. We started with one cell. At the end we have how many cells? At the end we have 2 cells. From one cell divide into 2 cell, but number of chromosome as well as number of DNA content is remaining the same. So can I call it equational division? Because there is an equal number of chromosome and DNA content in parent and offspring. There is no variation, there is no change. So can I call mitosis? Can I call mitosis, the entire mitosis, students, entire mitosis as equational division? Equational division. Equational division. Equational division. Done. Do you all of you truly understand now why it is called as equational division? Because here is the proof. Here is the proof for all of you. Here is the proof for all of you. Why it is called as equational division. Right here on the board. Equational division. Done, students? Done? All of you understood why it is called as equational division? Yes. Now, with that information out, mitosis is over. Mitosis is over, students. Mitosis is over right now. Mitosis is over. Okay. Mitosis is over right now. Okay. Now, students, did we learn here? Let me show you a small thing. Let me show you small here. Can all of you see this? Now, students, what did we learn with? Okay. We started with interphase. In interphase, we had nucleolus. We had the DNA here, chromatid. Yes, we had the chromatid. Did we have the centrioles here? Yes, we had the centrioles in the case of interphase. Okay, in the case of interphase. In the case of prophase, what happens, students? In the case of prophase, there was condensation of chromosomes. Condensation of chromosomes. Can you see the centrosomes are moving towards the corner? They are moving towards the poles. They are moving towards the poles. Okay. Now, in the case of metaphase, what happened? In the case of metaphase, this is early, this is late. Here is metaphase. See, early, late prophase. Early prophase, late prophase. Do you see in the case of your late prophase, do you notice that the entire nuclear envelope is disappearing? It is disappearing. Now, in metaphase, what happens? Can you see? In the case of metaphase, all the chromosomes are aligned at the center. Can all of you see that? At the center, aligned at the center. Now, what happens in the case of telophase? In the case of telophase, do you see that? Splitting of sister chromatids. The entire sister chromatids split open. Split open. That is the entire anaphase. Then what do you have in telophase? Can you see? In the case of telophase, that is what is happening is half of the chromatids are this side, half of the chromatids this side. Single stranded, single stranded, single stranded. Okay, single stranded. Right? And can you see here? Nuclear envelope is reappearing. It is reappearing. So that is the best part about the telophase. Nuclear envelope is reappearing by them. Reappearing by them. So that is the entire mitosis. So did all of you understand mitosis? If you understood, a quick thumbs up in the chat and thumbs up on the video also, and let me know in the comment section if you have ever learned this chapter like this in 3D format, DNA content wise. Let me know in the comment section also today later on. Okay. Now with that basic information aside, that is the mitosis over, can we start meiosis now, students? Can we start meiosis? Can we start? Can we start now meiosis now, all of you? Can we start meiosis? Tell me, it's so easy, right? This chapter is so easy. If you understand this chapter, it's such a easy chapter. Okay, and all of you know I make things easy by any chance anyway. Now, a question to all of you. In a basic sexual reproduction, let's say humans itself, there is a formation of sperm, there is formation of egg. The sperm and egg will fuse together to produce a structure called a zygote. This zygote will develop into an entire human baby eventually. Okay. Now the question to all of you, this particular structure called as egg and the sperm, right? Do you think they are produced as a result of mitosis? Tell me in the chat, true or false. Sperm and egg is produced with the help of mitosis, true or false? Tell me in the chat quickly. Yes, exactly. Everything is easy, right? Everything is easy with me. I'm also called as easy sir. Okay, tell me in the chat quickly. Is it possible that both of these structures are produced as a result of mitosis? Tell me quickly. Tell me in the chat. No. Let me give a small glimpse. What if there was only mitosis? Do you want to know? Do you want to know about a world where only mitosis existed? What would happen that time? Let me give a small glimpse. Small glimpse. Okay. Let's take I have male here and I have female here. They are producing, they are both of them are diploid organisms. We are diploid organisms. Diploid organisms. Male is producing the sperm, which is, let's say, if it undergoes mitosis, if mitosis, sperm is also going to be 2n. If this female is producing, let's say, egg, also by mitosis, if mitosis. Students, why am I repeating if? You know why? Randomly, someone will take a clip of this video and they will tell, oh, this sir doesn't know anything. This sir is telling mitosis here, this sir is wrong. So I have to be very clear on what I say. If it is mitosis, okay, it's going to be 2n. So what will happen, students? The zygote is going to be what? Zygote is going to be 4n. Zygote is going to be 4n. Now imagine this develops into a male who is 4n now. 4n. Similarly, here we have one more zygote which is also 4n, and this results into a female. Okay, and this female is also 4n. And when they produce the gametes and they fuse, do we get 8n? 8n. And this 8n will again become more and more, increase in number. So do you think this is actually sustainable? No. This could lead to a lot of complication over and over and over and over. So is there any solution? Yes, there is a solution. The solution is called as what? Meiosis. Why? Because in the case of meiosis, there is a change in number of chromosome. So what is happening, my students? In the case of meiosis, there is a change in number of chromosome. If an organism is going to be diploid, and if the cell undergoes meiosis, here diploid is going to become haploid. So there is a change in the number of chromosome. The number of chromosome is half. The chromosome number is becoming half here. Okay? Students, clear? That is why it's also called as reduction division. I'll get to that later on. Why it's called reduction division. Okay? Change in number of chromosomes is happening here. 2n is becoming n here. If there is 46 chromosome here, there will be 23 here. 23 here. Now I'll give you an overview. I'll give you the entire overview right now of meiosis. Listen to me very carefully, the entire overview. Smash the like button also, all of you. What is the... Students, 1 second. 1 second. Now I'll give you the entire overview. Okay. Now I'll give you the entire overview. Okay. Listen to me very carefully, all of you. Meiosis. The entire meiosis is divided into. Entire meiosis is broadly classified into 2 categories. Here we have interphase. Here we have, let's say, M phase or meiosis, M phase. This particular interphase is divided into 3 separate categories: G1, S, and G2. G1, S, G2. Done? G1, S, G2. Okay. Okay. G1, S, G2. Done? Now, what about M phase, or how is the division in M phase? Okay. This entire M phase is further divided into, students, listen to me very carefully, into 3 separate categories. 3 separate categories, entire M. Okay. can't draw straight line now. The first one is called as meiosis 1. Okay. Then we have meiosis 2. Meiosis 2. Okay. Between meiosis 1 and meiosis 2, there is something called as interkinase. Interkinase. Okay. Done. Now this meiosis 1 is divided into. This meiosis 1 is divided into P, M, A, T. Prophase 1, metaphase 1, anaphase 1. So prophase 1, metaphase 1, anaphase 1 and telophase 1 because it is meiosis 1. And students, remember very carefully, this is the meiosis which is actually called as reduction division. Why? Because in meiosis 1 itself, the 2n is converted to n. 2n is converted to n in the case of meiosis 1 itself. Okay. Now there is interkinase. Let me tell you, interkinase is similar to interphase. Interkinase is similar to interphase, but it is short-lived. And unlike interphase, in the S phase there is DNA duplication. Here, in the case of interkinase, there is no DNA replication. No DNA replication. No DNA replication in the case of interkinase. Clear? Now this meiosis 2 again is further divided into PMAT. But here we have right now PMAT 2, metaphase 2, anaphase 2 and telophase 2. 2. Okay? Clear. Now understand, students. Here, this particular meiosis 2 is like actually equational division. So can I tell meiosis 2 is similar to mitosis. It is similar to mitosis. That is, meiosis 2 is similar to mitosis. Meiosis 1 is reduction division, where the actual change in ploidy takes place. Okay, other than that, meiosis 2 is just like mitosis. So all you need to do is to understand meiosis 1. If you understand, this you already know. This you need to understand today, that's all. Remaining part of today's class. That is all you need to remember. Meiosis 1. Clear, students? Clear? Okay. Now students, understand very carefully. This prophase 1, this right here, prophase 1 is actually further divided. Yes, it does. This prophase 1 is further divided into multiple categories or multiple sub-segments. Okay. This prophase 1 is further divided, students. Let me tell you. This prophase 1 is further divided into something called as leptotene, zygotene, pachytene, diplotene, diakinases. Prophase 1 is further divided into 5 phases. Let zebra play during the day. L for leptotene, Z for zygotene, P for pachytene, D for diplotene, D for diakinases. These are the number of steps under prophase 1. Clear? Clear. Absolute cinema. Clear. Now, before we go further, let me read the lines of NCERT to reconfirm everything. Okay. Meiosis involves 2 sequential cycles of nuclear and cell division called as meiosis 1 and meiosis 2, but only a single cycle of DNA replication. That is, interphase is happening only once. Interkinase, no DNA replication. Meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Okay. Meiosis involves pairing of homologous chromosomes. This we will learn now. Okay. And recombination between non-sister chromatids of homologous chromosomes. This statement is very important. You will learn this in Prophase 1. End product. Okay. Four haploid cells are formed at the end of meiosis 2. Meiotic events can be grouped under following phases. That is, these are the different phases right here. Meiosis 1, meiosis 2. 1, 2, 3, 1, 1, 1, 2, 2, 2, 2. These are the different phases under prophase 1. Leptotene, zygotene, pachytene, diploidine, diakinesis. So with that basic information about meiosis, can we start meiosis 1, prophase 1, first stage called as leptotene? Can we start, students? Can we start now? Can we start? All of you. The first phase under meiosis 1, that is prophase, leptotene stage here. This stage right here, this stage, leptotene. Okay, very simple, very simple. So what exactly happens in the case of leptotene? In the case of leptotene, the chromosomes becomes gradually visible under light microscope. That's all. Okay, that is first stage in prophase, first stage. Chromosome gradually become visible under light microscope. Under light microscope. Under light microscope. Okay, compaction of chromosome continues, followed by zygotene. That is all. 1, 2, 3, 4. Write down, students. Write down. Okay, write down. Okay, done. Now, what exactly happens in the case of zygotene? Now there is something called as homologous chromosomes. Now, how do you understand this concept? Now, students, in a school, right, in your school, generally in your class, usually when they ask you to make a group project, or let's make a group of 2 people, whom do you choose? Do you choose your friend who is similar to you, who thinks like you, who behaves like you, or do you choose a random person from your class? Tell me in the chat. Whom do you choose? You always end up choosing who is similar to you in your group to make your friends or to do the project also. Yes or no? Yes. If you are smart, you'll go with the topper. Okay, because they will do all the work. Okay. So what do? You make a team with your best friend who is similar to you. Now in zygotene stage, we are actually making a group right now. We are making the group at the zygotene stage. So what is happening here is, inside the cell, there are many chromosomes everywhere. Okay, inside the cell. 2 chromosomes which are called as homologous chromosomes because they are alike. They look like alike, they behave like, okay, same. These homologous chromosomes will start to come together and they will pair up. This process of pairing of homologous chromosomes, this entire process is called as what, students? Synapses. Okay, it is called as synapses. For example, all of you follow right now. Here I have 1 chromosome. Let's take here this chromosome and this chromosome. Both of them are homologous to each other. Yes, they are. And they will pair up. They'll pair up like this. This pairing up is called as synapses. It's called as what, students? Synapses. Now understand here, between the chromosomes, there is a complex formation between the chromosomes. Now this is called as synaptonemal complex. This right here is called as what? Synaptonemal complex. It is called as synaptonemal complex formation. Okay. Now students, understand, this stage right here is called as bivalent it is called as bivalent, or it can be called as tetrad stage. Tetrad stage. Bivalent or tetrad stage. This one here. Now, why is it called as bivalent? How many chromosomes? Tell me, students. How many chromosomes? One chromosome, second chromosome. So two chromosomes, bivalent stage. The same stage is also called as tetrad stage. Why? How many chromatids? Tell me. 1, 2, 3, 4. 4 chromatids. It is called as tetrad stage. Tetrad. Now listen to me very carefully. Now this formation of bivalent and formation of tetrad starts at zygote. Starts at zygote. But the completion happens in the next stage. I'll repeat again. Formation coming together is called as synapses. There is formation of synaptonemal complex. Third main point, this entire formation of bivalent tetrad starting is in zygote, but ending is in where? Pachytene. So bivalent chromosomes clearly appear as tetrad. So the clear distinction or the clear appearance is happening in the case of Pachytene stage. In the Pachytene stage. Okay. It is longer than the previous stages. Pachytene stage is much more longer compared to your leptotene and zygotene. Okay. Now what exactly is happening here, students? Here there is a process called as crossing over, which I want everyone to understand here. Crossing over. What is crossing over? Okay. What exactly is the meaning of crossing over? Now students, I want everyone to focus here. These two are called as what? They are called as sister chromatids. They are called as sister. Now what about one from here? Okay. Now what about one from here and one from here? They are called as non-sister chromatid. Non-sister chromatid. Now there will be something called as exchange of genetic material. Exchange of genetic material between non-sister chromatid. This will go here, this will go here now. That's all. Okay. Now understand. This is also non-sister. Okay, you can make these two also non-sister. Okay, non-sister. I've just mentioned this one. Both of them are also non-sister. Now what is happening here? Understand. Okay. First of all, there is something called as crossing over. Okay. Now till here, it's very easy to understand. Very easy to understand. Okay. Now what happens? You understand. I'll write right here. 1. 1, 2, third diagram. Can all of you notice in the third diagram what is happening in the third diagram? Can all of you notice what exactly is happening in the third diagram? This chromatid which is here is gone this side and this has come this side. There is a complete exchange of chromatid is happening here. The pink has gone this side and the yellow has come this side. That right here is called as crossing over. What is crossing over? Site of crossing over between non-sister chromatids. Crossing over means what? Exchange of genetic material. Exchange of genetic material between two homologous chromosomes of non-sister chromatids. So crossing over is happening here. That is exchange of genetic material between non-sister chromatids of homologous chromosome. Now here at the site of crossing over, at the site of crossing over, there is a formation of something called as recombination nodule. Recombination nodule. There is formation of recombination nodule. And this entire thing is enzyme mediated, and the enzyme here involved is recombinase enzyme. What is involved here? Recombinase enzyme. Recombinase enzyme is involved here. This is the enzyme involved here. So this is called as crossing over. Exchange of genetic material has occurred here. Exchange of genetic material between non-sister chromatids of homologous chromosome. Homologous chromosome. And remember, students, between them the synaptonemal complex is still there. It is still there. It is still there. Okay. So done. 4 main points. First point is they appear clearly bivalent. Second point is crossing over. Third point is recombination nodules. Fourth point is recombinase enzyme. Now what happens? Can you see here? Exchange of genetic material has happened here. Okay. Next up is called as diplotin. Now in the case of diplotin, what happens? Understand all of you. Okay. Now I can't copy this. Okay, I can't copy it. Let me draw it one more time. Now students, can you see this synaptonemal complex in the middle? This synaptonemal complex, this entire synaptonemal complex will do what? Disappear. So first step is what? The dissolution of synaptonemal complex. So the entire synaptonemal complex will disappear. Now, recombined homologous chromosomes separate. They will completely separate out. They will separate out except at a point of crossing over. Can you see here? This is called as point of crossing over. Except at the point of crossing over, they will not separate out. Okay. Now this point of crossing over will produce a X mark. Okay. This X mark is called as chiasmata. This X-shaped mark is called as chiasmata. So what is happening here at diplotin stage? The synaptonemal complex is dissolution. 2 chromosomes will separate out except at the X mark. That is, X mark is called as chiasmata. Okay. Now last stage is called as a diakinesis. In the case of diakinesis, what happens is the X-shaped mark is there, right? That X-shaped mark also terminates. That is called as termination of chiasmata. Chromosomes condense fully because this is prophase. So entire chromosomes becomes condensed because prophase means condensation of chromosome. Okay. Nuclear envelope disappears. Nuclear envelope breaks down completely. Represent transition to metaphase. So prophase is done. Let me give a small recap what we learned right now. We learned that in the case of leptotin, chromosome becomes gradually visible under light microscope. Zygotin, there is formation of synapses, synaptonemal complex, bivalent tetrad. This bivalent tetrad is more clearly visible in the case of pachytene. There is crossing over, there is recombinase enzyme involved, and there is formation of recombination nodule at the site of crossing over. Then we learnt about your diplotin, that is dissolution of synaptonemal complex. Then there is X-shaped mark called as chiasmata. Last step is called as diakinesis. There is a termination of chiasmata. Chromosomes become completely condensed. Nucleolus disappears. Golgi complex disappears. Everything is disappearing. Ready for metaphase now. Ready for metaphase. Okay. Now we come to metaphase 1. Metaphase 1. Now the difference here in the metaphase 1 is very evident, okay. For example, this is a cell right here. This is a cell. In the case of normal mitosis, we had something like this, yes or no? In the case of normal mitosis, did we have a single equatorial plate? In the case of mitosis, metaphase. But this is meiosis now. In the case of meiosis, there is homologous chromosomes. Yes, there is homologous chromosomes. So we have two metaphasic plate. We have two separate metaphasic plate now, because this right here is going to be metaphase 1 of meiosis. Because in the case of meiosis, there is homologous chromosomes involved. Now here, again we have kinetochore. All the spindle fibers will attach to the kinetochore. But the difference here is one spindle fiber is attaching to one chromatid, not both the chromatid. This side chromatid is free. This side chromatid is free. Free. Other side, attach. So here what is happening here is spindle fiber is attached to only one chromatid. Only one chromatid, not to both of them. It is attached to only a single chromatid. Now students, you want to tell me logically, if I pull from this side and if I pull this from this side, will the centromere split? Obviously not. This entire homologous chromosome will come down. This entire set will go up. All of you know. That is what is happening in the case of anaphase. In the case of anaphase 1, homologous chromosomes separate. So centromere is not splitting. Sister chromatids remain attached. They remain attached to each other. There is separation of homologous chromosomes. That's all. Okay. See, homologous chromosomes separated. The entire chromosome is being pulled. Can you see? The sister chromatid is still there. They're still attached. Centromere is still there. They're being pulled apart. Now, telophase 1. Nucleolus, nuclear membrane, everything is reappearing. This was one cell. This is how it looks at the end of. Telophase. Everything is reappearing. Nuclear envelope is reappearing. And here we have something called as dyad of cells. It's called as dyad of cells. Okay. It is very short-lived and there is no DNA replication. After this, we get two cells. That is cytokinesis happens here. Cytokinesis. Okay. Now from an entire diploid cell, we are getting two haploid cell. That is, one diploid cell gave rise to two haploid cell. This right here is called as a dyad of cell. It's called as a dyad of cell. We are here right now. Telophase 1. Now, can you see? This right here is telophase 1, students. Telophase 1. Clear, students? Clear? Clear, all of you? Clear. Telophase 1. And let me tell you, students, this cell right here is N2C and this also is N2C. We'll come back to this. N2C. Why it is N2C also. Okay. N2C. Okay. Now, interkinase is short-lived. No DNA replication followed by prophase 2. Prophase 2 starts here. Now, from here, everything is easy. All of you know prophase 2. What happens? Condensation of chromosomes happens. That's all. Condensation of chromosomes, nuclear membrane disappears, chromosome becomes compact. Prophase 2. Metaphase 2, same thing again. Metaphase 2, all the chromosomes align at the equatorial plate. Just like mitosis. What is happening? In anaphase 2, what is happening? Centromere of the sister chromatid is split. They're splitting open. So if I ask you a question, right? In which of the following stages of mitosis and meiosis Sister chromatid separates. What is your answer? Tell me. Crazy Dude, Roshni, Ravindrani, Yeshu, Meghana, Yalank, Yalia, Kubera. Tell me. The answer is very simple. In mitosis, anaphase. Meiosis, anaphase 2. Meiosis, anaphase 2. Mitosis, normal anaphase. But in the case of meiosis, it is anaphase 2. Anaphase 2. Clear? In both of these situations, both of these phases, the centromere splits. Centromere splits. Telophase 2 again, very simple. In telophase 2, nuclear envelope again reappears. Formation of tetrad of cells now. Now there is formation of tetrad of cells. Tetrad of cells. Okay. That is entire 2n gave rise to n. This was diad. These two cells underwent again. This till here it was meiosis 1. From here to here it is meiosis 2. And we end up getting 4 haploid cells. We end up getting 4 haploid cells in the total. 4 haploid cells. Okay. Significance, conservation of chromosome number. Obviously now chromosome number is maintained. Genetic variability, evolution. That is, because of crossing over, there is variation. And variation can lead to evolution. Okay. Lead to evolution. Now students, this is a telophase 2. Okay. This is telophase 2. And this each cell is NC. It's just NC. How I'll tell you right now. We come to almost towards the end of today's class. That is DNA content for meiosis. Ready all of you? Ready to understand it better now? Can we start? Students? DNA content for meiosis now. DNA content, entire calculation for meiosis now. Entire calculation for meiosis. Can we start? All of you? In 2.5 hours, the chapter is over in detail with animation, with DNA content. All of you drink some water now. Quickly drink some water. Done? Do you realize how the chapter got over so fast? Do you realize how the chapter actually got over so fast? The chapter got over. Such an easy chapter. Okay. Now students, let's take a human cell. How many chromosomes? 46 chromosomes. 46 chromosomes. Let's take a DNA with 40 picogram. 40 picogram DNA. Okay. Here, let's write down stages. Let's start down stage. Okay. Number of chromosome. Okay. Number of chromatid. Number of chromatids. DNA content. Okay, DNA content. Okay. I think this much I can pull. Now just copy the slide so we don't have to draw it again. Okay. No students. All of you start. Let's start. Let's start with G1. G1. Number of chromosomes, tell me in the chat quickly. We'll have 46 chromosomes, 46 chromatids. DNA content is going to be, let's take DNA content is going to be G1. No, G1 is going to be 40 picogram. 40 picogram. Okay, 40 picogram. Let's take S. Number of chromosomes is going to remain the same. Chromatid is going to be doubling now. Doubling. 92. This is going to become 80 picogram, 80. Direct content doubles. G2, G2 is going to remain the same. What if I say, students, all the way from the G2, right? G2 is going to remain the same. Metaphase is going to be the same. Prophase, metaphase, anaphase, it is going to remain the same to same. Only in telophase there will be a small change here. Okay. In telophase there will be two things which you need to understand in telophase here. Telophase, two things. Okay. Now here in telophase there is something called as per cell and per nuclei. Per cell and per nuclei. Per cell it is going to remain 46, 92 and 80. When I talk about per nuclei, it is going to become completely half. Okay, it's going to become completely half. Let's say this is going to become 23. It's going to become half. This is going to be half to 46. This is going to be half to 40. How, sir? Why, sir? Okay, listen to me. Let's come back to telophase. Telophase 1, telophase 1. This is the entire cell here. This is the entire cell here. It has 2n, 2n. Okay. 2n. Okay. This right here is 2n right here. Okay. 2n 4C condition. Okay. Here we have, let's say, 23 chromosomes. 23 chromosomes. 23 chromosomes are there. Okay. Better image there. No, better image was there. Okay. Now this entire cell in the telophase, entire cell is 2n 4C. Same thing is continuing. From G1. From G1 till here, it is 2n 4C only. 2n 4C. Unlike in the case of mitosis, it became 4n 4C because of the chromatid becoming like chromosome in anaphase. That does not happen in the case of meiosis 1. Does not happen. Okay. It is going to remain the same. Now from this 2, 4 and 4C, half will go this side and half will go this side. Yes or no? So can I say half of 2n is what? N. Half of 4 is what? 2 here and 2C here. So half 23 chromosomes this side, 23 chromosomes this side. Every chromosome has 2 chromatid. They have 2 chromatid. Listen to me. They have 2 chromatid here. So 46, 46. Come back here. Listen to me, all of you. This is the entire cell. 2 and 4C, 2 and 2C became 2 and 4C. Here, 2 and 4C. Same 2 and 4C was continuing till here, 2 and 4C. Now this 2 and 4C here, right? 2 and 4C split into 2 types per cell, per nuclei. So it became N, 2, C here, half and half. So this entire cell, if there was an entire cell in telophase, 23 chromosomes went this side, 23 chromosomes went this side. Entire cell is 46. Per nuclei it is 23. Okay, 23. And this every single chromosome has 2 chromatids because there is no splitting here. It is meiosis 1, right? Meiosis 1. There is no splitting of centromere, right? So 46 chromatids went this side, 46 chromatids went that side. Okay, so 23 will be this side, 23 will be that side. That's all. This entire thing is for meiosis 1. This entire thing for meiosis 1. Now what happens in meiosis 2? In meiosis 2. Okay, so meiosis 2 now. So meiosis 1. Now what happens in the case of meiosis 2? Okay, let's take this cell itself, N2C, right? N2C. Okay, what do we have? 23, 46, 40. Okay. Stage is going to be, let's say, prophase 2. Okay. Number of chromosomes is 23. Number of chromatids is 46. DNA content is 40. This is N, 2C. Okay, this is G1. Okay. Now what happens in the case of S phase here, students? Tell me, what happens in the case of S phase? Will there be S phase here? No, there will be no duplication. Here in the case of meiosis 2, there is no duplication of DNA. Remember that. In interkinesis, there is no duplication of DNA, so it is going to remain the same. Now come back to G2 is going to remain the same. Prophase also is going to be the same here. 23, 46, 40. Prophase, there is no change here. Remember, here in S phase there is no duplication. I wanted to repeat it one more time. Interkinase, no duplication again. Okay. Now, what happens in the case of anaphase here, special anaphase? In the case of anaphase, this anaphase 2 is similar to mitosis. Anaphase 2 is similar to mitosis. And in the case of mitosis, we learned that every single chromosome is acting like every single chromatid is acting like a chromosome. So can I tell this entire chromatids will act like a chromosome here? 46. DNA content will remain the same. 40 will remain as 40. Now what happens again in telophase here? Per cell, per nuclei. Okay. Per cell it will remain the same. 46, 40. What happens per nuclei? This entire 46 chromosomes is going to go this side. Half of them will go above. Okay. See here. This entire telophase 2 is 46 chromosomes. Is 46 chromosomes. Out of these 46 chromosomes, can I tell half is going this side and half is going this side? Okay. And this right here was N2C. But as soon as we come to anaphase, what happens, students? As soon as we come to anaphase, 40, it became, as we come to anaphase, 23 became 46. So at anaphase, temporarily we have 46 chromosomes. So here anaphase it is 2N, 2C. Because it is N here, it is because 2N here. N2C. Okay. Now, nuclei, you understand now what happens. 2 and 2C is the entire cell, even in the telophase per cell also. Okay. This entire cell is 2 and 2C. Half of this N is going to go this side. Half of this C is also going to go this side. Half of this N will going to go this side. Half of this C will go this side. So can I tell N is going this side? N. Half C is going this side? Only C. Half N is going this side, half C is going this side. So NC and NC. So what do we have here? This will become per nuclei. This is going to be 23. This is going to be 23. And this is going to be half of is going to be 20. This is going to be NC now. Half and half gone this side. Okay. So make it into half per cell per nuclei, NC. So we started with what? We started with 40. In G1, we started with 40. In G1, one second. In G1, we started with 40 picogram of DNA. 40 picogram of DNA in G1. But at the end, what do we have? 20, 20, 20 and 20. We have 4 different cells. Now, how do we have from 40, 20, 20, 20? Doesn't make sense. 20, 20 plus 20 is what? 40, 40, 80. So, how do we get from 40 picogram to 80 picogram? How? Because In the S phase, it became 80 picogram. And this 80 picogram is divided into 20, 20, 20, 20. Okay, 20, 20, 20. That is the magic here. Okay, that is the magic which is happening here. 20, 20, 20, 20. That is, in the S phase it is dividing in the S phase. Okay, so that is the basic of meiosis. Okay, let me give a recap of one more time, students, because all of you, a little confusing it might be. So listen to me. What happened here? First we had human cell. Okay, human cell, 46 chromosomes, DNA count 40, meiosis 1. Meiosis 1. Okay. G1 46, 46, 40 DNA, 2 and 2C. 2 and 2C. S phase became double, double, 2 and 4C. DNA count became double. Until G2, M, P, prophase, metaphase, anaphase is going to remain the same itself. Is meiosis 1 remain the same itself. Per cell is going to remain the same. But per nuclei, it is going to divide into half now. Divide into half. Because out of this 46 chromosomes, 23 is going up, 23 is coming down. Reduction division. This is a reduction division. So half is going this side, half is coming this side. 23 here, 23 here. Okay. So half chromated, half. So 80 became 40, 2 and 4C became, half of this is N, half of this is 2C. This is after meiosis 1. Now if you take the same thing, N2C, N2C same thing. Okay. In S, there is no change here, no DNA replication at all. Okay. Prophase, nothing. But anaphase, again there is a change. Why? Because anaphase 2 is similar to mitosis anaphase. Mitosis anaphase and this anaphase is same. It is same. Okay. So here what is happening is, this entire chromatid is acting like a chromosome. So chromosome number is gonna double. Like how we had 4 and 4C there. 4 and 4C, same doubling is happening here. Doubling. So this N2C is becoming 2 and 2C. Chromosome number is temporarily doubling. Per cell remain the same. Per nuclei, again half. 2N became N, 2C became C. So we have NC here, and here we had 2 and 2C. So from one cell we got 4 cells. From one cell we are getting 4 cells. I'll repeat again here. We had diploid cell. We got 2 cells NN, and these 2 cells also became N and N. Here it is meiosis 2. Here it is meiosis 1. Meiosis 1. And we get 4 haploid cells. Okay, 4 haploid cells. Clear, students? Clear? Now I know this might be too much to take at one go. I want you to watch the lecture one more time and write this and practice. Write this entire thing in practice. If you have any doubt after this also, you can always let me know in the comment section and I will clear it for you. Okay, that is my guarantee. Okay, so thank you so much, students, for today's class. It was really fun, and I believe all of you made the target. I don't know, I didn't check my target. Okay, so like the video, let me know in the comment section how was the 3D part of this chapter. Okay, and students, let me know in the comment section if this was helpful to you. This entire PDF will be available in Telegram channel and also in the link in today's video. In today's video, I will make sure the link is there, so it will be uploaded there itself. Okay, so thank you so much, students, and I will see you in the next class. I hope it was very productive class. All of you who have backlog, I hope it was clear to all of you, and thank you so much, and I'll see you in the next lecture. So bye bye, take care all of you, bye bye. Okay, boy, both PDF I will send you, don't worry.